So auxin is a plant hormone that helps the plant respond in a variety of ways to different kinds of environmental stimuli. For example, auxin can help with either the movement towards a stimulus, which is going to be positive tropism, or away from the stimulus, which is going to go ahead and be negative tropism. We're going to go ahead and look a little more closely on the next couple of slides to see how plants move either towards or away from light, which is going to be phototropism, towards or away from gravity, which is going to be gravitropism, or towards or away from touch, which is thigmotropism. So a quick reminder, we're going to be using the word tropism when we're referring to plants, but taxis when we're referring to animal behavior. So make sure you keep your vocab straight. So here we're looking at two experiments. So we're going to look at this one first. Of course, you're going to need a control to observe what's occurring under normal circumstances. And in this case, you're going to see that normal circumstances is going to involve having light, okay? And then the grass is going to bend towards the light. But if you remove the cap, if you remove the tip, or you go ahead and block the tip in any way with an opaque cap, you're no longer going to get that response and the grass is going to stay straight. But if you have a transparent tip, so light can still penetrate, or you only block the bottom of the grass, you're still going to get a normal response. So what's going on here? So number one, the experiment is showing us that this response is somehow related to the tip of the plant receiving that light signal. And two, we now know that the hormone is auxin, and that's located at the tip of the grass. And it's that hormone that's going to increase the flexibility of the plant's cell walls, but it's only going to do that on one side. So the plant cells that are further away from the light are going to experience a greater auxin concentration. So you're going to get more auxin build up here on basically the dark side of the grass. And that means that you're going to go ahead and elongate those cells, and it's going to cause the shoot to bend towards the light. So that mechanism is allowing the plants to optimize the amount of light that they're receiving in order to increase photosynthesis. And that's just an example of positive phototropism. So in the next experiment, we're going to see that when the tip is removed and then a gelatin block is placed between that and the shoot, you're going to see a very different response if the gelatin block is permeable versus if that's happening with a gelatin block that is not permeable. So in the case where it is permeable, auxin, which is located in the tip again, can go ahead and diffuse from high concentrations in the tip to low concentrations in the shoot, and you're still going to get that positive phototropic response. But when the block is not permeable, those auxins that are concentrated here in the tip cannot flow to the shoot, and you no longer get a response to light. So now we're going to look at gravitropism. Plants can detect gravity by statoliths, which are these cytoplasmic components that settle wherever gravity is pulling them. So even if a plant is placed on its side, the plant is always going to know where up and down is. And based on that, the auxin concentrations are going to accumulate, and they're going to stimulate different plant tissues to behave differently. So for example, in the roots, auxin, when it goes ahead and starts to accumulate, okay, it's going to inhibit the growth of the lower cells, and it's going to go ahead and elongate the cells on the upper section. And then it's going to cause those roots to curve downward. And that's going to exhibit positive gravitropism in the roots. And on the other hand, in the shoots, when you have the auxins go ahead and accumulate here, it's going to activate cell elongation on the lower cells, and then that's going to cause the plant to bend upwards. And that's going to exhibit negative gravitropism because it's going against gravity. And remember, thigmotropism is the directional growth in response to touch. In this case, auxin can cause the cells of the stem to elongate and curve when touching a surface. And that allows the plant to coil for support and climb taller and then potentially even reach more light. That's an example of positive thigmotropism. So now we're going to look at two different hormones. One of them is ABA. We're going to start with that. That's abscisic acid. And that's going to help the plant in terms of drought tolerance. So ABA can be released in dry conditions. And that's going to be important because it affects how the stomata is going to behave. So when the ABA is released and it goes into the guard cells, it's going to go ahead and cause the potassium ions to rush out of the stomata. And that pulls the water out with it, which is going to go ahead and make those guard cells flaccid, and it's going to close the stomata and allow that plant to kind of conserve water. That's why it's helping with the idea of drought tolerance, right? It's going to trigger the stomata to close, and it's going to do that with that osmotic potential. On the other hand, you can obviously reverse it, and then if you remove that ABA, so if you take the ABA out, the potassium is going to go ahead and rush back into the guard cells, and then you also have water rushing in. That's going to make the cells turgid, and then you're going to open the stomata. But abscisic acid also has a role in seed dormancy. So when it's present in seeds, they're going to say, stay dormant. So in this case, our ABA is right here, and it's located in the seed, and it's going to go ahead and antagonize the actions of any type of growth hormones. However, if you end up having a really large amount of water, so the plant could potentially grow really well, when water is released maybe through a big rain, it's going to go ahead and wash that ABA right out of the seed, and that's going to go ahead and start allowing gibberellins to go ahead and do their job. And these guys can play a role in stem elongation, fruit growth, but we're really going to focus on the seed germination aspect. So after the water is added, right, and the ABA is washed away, you're going to go ahead and release these gibberellins, and it's going to go ahead and break the seed out of its dormancy. And the enzymes are activated, and they're going to start digesting these starches so that the plant can start using more energy for respiration to start making shoots and roots. So cytokinins are a class of growth substances that promote the idea of cell division. They also help with differentiation, so the plant can go ahead and have different types of tissues. Cytokinins are also involved in maintaining the meristems. And meristems are the tissues in most plants that contain undifferentiated cells, meristematic cells. And they're going to be found in zones of plants where growth is going to be taking place. And those meristematic cells can give rise to a various amount of plant tissues. And again, they're responsible for growth, and then cytokinins play a major role in that. Our last hormone that we're going to look at is ethylene, and this is going to be a gaseous hormone, and it's going to be involved in positive feedback. So when you have this ethylene gas, it is responsible for, again, breaking down the cell walls of the plant. So when you go ahead and start taking those cell walls and you break them down, the fruit is going to get a little softer. It's going to get a little riper. It's going to start smelling really good, and it's going to go ahead and produce more ethylene gas. And then that ethylene gas is going to go ahead and cause the cell walls to break down even further, which is going to cause you to make more ethylene gas, which is going to go ahead and cause you to break down the cell walls even further. So that is a positive feedback loop there, and ethylene is involved. So now we're going to go ahead and look at phytochromes. And they consist of two identical proteins that are joined to form one functional molecule. So the important thing to know here is there can be a conversion back and forth between the two forms. So the two forms are PR, which is going to be the inactive form, and then there's PFR, which is the active form. So when you're inactive in the PR form, when there is red light present, so in the daytime those levels are going to go ahead and rise, those red lights are going to go ahead and trigger the active form of the PFR. But again, you can kind of go back, you can convert when darkness starts to kind of take place and the light levels start to reduce, you can go and switch back to the inactive form of that phytochrome, the PR. So in that way, the plant knows the daytime from the nighttime, and they're going to have circadian rhythms just like we do. So in terms of what's going on with a shaded area here underneath the tree is going to screen out more of the red light, and it's going to leave the far red light for the plants below it. So the phytochrome ratio is going to shift more to a PR version, and it's going to change the growth pattern of those plants, and the plants are going to start to elongate their stems to try to reach the light rather than producing a bunch of healthy and bushy branches where it's really sunny. So here we're going to go ahead and link this idea of phytochromes to what we've done in the last unit, which was signal transduction pathways. So in this case, what we're looking at is in a potato, right? This down here is supposed to be my potato. You've got these ice buds coming off, and you're growing shoots, and that little potato is trying to grow these shoots to basically find nutrients, and it's trying to do that before the tuber dies. So if light ever does end up hitting that potato, it's going to go ahead and activate the phytochromes. So that would be stage 1, reception. And then you're going to have stage 2, where you have signal transduction pathways. And in the end, what you're going to have happen is de-etiolation. So this concept right here was etiolation, when you have those shoots growing. But again, if it reaches the light, it's going to go ahead and have de-etiolation take place, and you're going to start greening the plant, and it's going to start making leaves to help it photosynthesize. So we're also going to be looking at this idea of photoperiodism. So it's going to be this environmental stimulus that the plants can use in terms of light to have a physiological response. And in this case, we're looking at the flowering. So there's going to be short day plants and there's going to be long day plants. So short day plants, like the name implies, are only going to flower if the days are short and the nights are long. If the days are long and the nights are short, it's not going to go ahead and flower. So short day plants flower only when the period of darkness exceeds this critical night length. Long day plants, like the name implies, is going to be a little bit different. So they are only going to flower when the day is long and the night is short. And if the night is long, you don't get any flowering. So the interesting thing is, if you hit the short day plant, right, under normal circumstances, this looks like it should flower. But if you hit the plant during the nighttime with a flash of light, you're going to go ahead and reset those phytochromes, and then the plant is going to interpret that as a long day, and it is not going to flower. And the same is true for long day plants, if you're going to go ahead and take those guys. So again, under this circumstance, what should happen is this plant should not flower. But if you hit it with a flash of light, again, you are telling the plant, oh, hey, it's actually a long day. So if that's the case, yeah, let's flower. So here we can look at, I'm going to focus right here on the short day plant. You can go ahead after the video is over and maybe look at the long day and make sure you understand it. But for the practical idea of time, let's just focus on one. So just like in the other slide, if you're going to have a short night and a long day, if it's a long day plant, it is not going to go ahead and flower. It needs a long night and a short day to go ahead and flower. But the interesting thing is, if you hit it with a flash of red light, it's going to go ahead and shorten the dark period. And if that's the case, if the plant goes ahead and has or perceives a shortened dark period, it's going to resemble the first example where we looked, and that resembled more of a short night and a long day, and you're not going to get any flowering. But if you hit it with a subsequent flash of far red light, so here, right? So you hit it first with red light, then you hit it again with far light. That can cancel out the red light's flash effect, and you're going to go ahead and reset it, and the plant is going to perceive that as a long night and a short day, and it is going to flower. So every time you hit the plant with a flash of light, whether it's far red light or just red light, that photoperiodic response is going to reflect whatever it was last hit with. So in this case, it doesn't matter how many times you're hitting this. It starts here, red, far red, red. If it's hit at the end with this red light, okay, it's going to go ahead and resemble what it did in the short night, long day plant, and it won't flower. And again, whatever you hit it with last, it's going to go ahead and perceive that is far red light will be then for flowering.